Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Ashleigh Wake from Intertek.
Hi, thank you, Leah, very much for the introduction and for everyone for tuning in today. It's my pleasure to be here. Before I move on to talk about the main bulk of this presentation, what I'd like to do is just to give you a really quick introduction to Intertek, really to just give you the context to where we're coming from by delivering this presentation. So Intertek is a very large global organisation, the one that often people have never heard of. We operate over multi-different industries. However, our centre of excellence for the analysis of complex and biological therapeutics lies in Manchester in the north of the UK. And this site has worked with biologics, including peptides, for over 20 years. And we have a wide range of technology and expertise which we use to help our clients design and deliver the very best strategic solutions for their power peptide characterization, stability and quality control programs. And I really want to stress in that is the word strategic, as we never look at this type of work as kind of simply ticking a box to meet a regulatory expectation, but very much as something which we define behind the science of the product and its application. So to very quickly recap on the kind of peptide therapeutic market, we know that peptide therapeutics are not a new invention, but they've been around really since the advent of insulin in the 1920s. And since then, there's been a real steady growth in the market in the industry, which has been compounded in recent years with the emergence of generic versions of marketed products such as glucagon and liraglutide. There is massive potential for this class of markets given their wide range of application and therapeutic areas. Advances in target identification, synthetic and cellular routes of manufacture, and importantly in the context of this presentation, improvements in analytical methodology have and will continue to have a really big part to play in driving this potential to marketable products. When we start to think about what actually drives our product to registration, and in terms of where peptides lie in a regulated space, it's often quite difficult to describe because in many ways they kind of fall into what could be kind of thought of as a regulatory vacuum, and they're completely aligned to small or large molecule guidance. One thing that can really be agreed, however, is the definition of what a peptide constitutes. And when we're talking about a peptide in kind of all these regulations and in all our kind of discussions, we're really talking about a polymer composed of 40 or fewer amino acids, or by rule of thumb, something of 5,000 Dalton mass or less. So in the absence of any defined guidance, when putting together strategic phase-appropriate solutions for monitoring the quality of peptides, it's really imperative to actually consider the inherent characteristics of the peptide, and that was that through suitable analytical control can continue to establish that the product remains efficacious, and safe both at the point of manufacture and throughout the product's shelf life. So when we stand back and we think about all we know from kind of historic evidence and literature about what affects the safety and efficacy of a peptide, 4 distinct areas of concern are really highlighted or really jump out. And these specifically relate to the peptide structure, their physiochemical properties, their impurity profile, and the interaction they have with excipients and containers in the final product. Using this knowledge as a basis, the remit of what a QC program needs to look like and a testing requirement becomes much easier to define. And the strategy is then based on systematically working through what each means in terms of peptide analysis and what is appropriate to each phase of development. The word comparable or consistent is used a lot when describing quality control programs, because really the essential aim of any program is to ensure that each batch of material produced is the same as the batch used in preclinical and clinical studies to confirm the safety and efficacy. Where we can't prove identical, what we need to do is really define the limits of acceptance for differences that we might see. The first layer of structure and the first point we need to consider is around the structure of the peptides. And the first layer really relates to the primary sequence of the peptides, or the identification and the sequence of the amino acids that actually compose the peptide. Elucidating the overall distribution of amino acids, the sequence, and the stereochemistry is a really key quality control attribute. And this is needed really not just to tick any box for identity tests, because the primary sequence of the self and any modifications can lead to real changes in efficacy and safety, and it's imperative to know this so we can avoid it and monitor it. So if you imagine when choosing a peptide to take into development, modelling may have indicated tens of different sequences with a potential desired mode of action or effect, and many of these will have been quite similar in length as a peptide and even composition, but many will have been rejected because of their immunogenicity potential, which can be modelled in suitable programs. So what this really kind of drives through is that small changes in sequence can have a really significant consequence. And so in any characterization program, extensive work really has to be done to confirm this primary sequence and any modifications that may be present. And this is true for really early batch release programs as well as characterization. But the more and more is understood on batch-to-batch consistency, the need for full sequencing on a routine basis may in fact reduce and be replaced with a similar ID test such as intact mass, and then the full structural analysis only performed on selected batches. Amino acid analysis, we use one of the many chromatographic or amino analyzer type approaches in line with European, US, and global pharmacopoeia compendial methods, are the best approaches for establishing the quantitative distribution of amino acids in a sample, and also can be used to derive overall peptide concentration on the basis of well-recovered amino acids. However, they don't provide any information on the actual sequences of the derived sequence. Evaluation of this is most effectively assessed using LC-MS, which may include for larger peptides a protease digest to actually break the peptide itself into smaller peptides suitable for MS, high resolution or MS-MS sequencing. Similarly, Edgeman is a powerful technique which sequences amino acids from the N-terminus and provides data similar to LC-MS, and is that way is really an ideal confirmatory tool. However, these two techniques don't only confirm each other and add kind of confidence in the data, but they also each has additional benefits, such as the ability of Edman to distinguish between isobaric amino acids, such as leucine and isoleucine, and the additional information that can be gleaned from LC-MS on post-translational modifications, or importantly, disulfide bridge mapping, which is an incredibly important indicator that the right higher order structure is being formed. As well as primary structure, peptides display high conformational flexibility with secondary structure that is on the main restricted to alpha helixes and beta sheets with the remaining unordered structure. Changes in secondary structure directly influence the activity but also safety of peptides. Analytically wise, there are several methods which can be used to estimate secondary order structure. These include circular dichroism, FTIR, Raman, NMR, and X-ray crystallography, to name but the main. Each of these methods, again, has its own benefit and plus point. However, certainly during characterization and early batch release programs, the use of orthogonal approaches is very much required to ensure a full and quality data set. The combination of testing chosen should really complement each other. And one really strong combination that is used quite frequently is CD and FTIR, which produce complementary data on the percentages of alpha helix, beta sheets, beta turns, and an ordered structure that might be present. You can use these data if it's comparable between the two techniques. You have a lot of confidence that you're actually looking at something real and complete. If, however, you see differences, that really gives you the kind of opportunity to investigate further and maybe introduce a further technique such as Raman or NMR. A standalone analysis, NMR and X-ray crystallography, are the only technologies that will actually allow us to get information simultaneously on the whole structure. So therefore, these type of analyses provide really a high level of structural evaluation. And when you compare the spectra that they generate to that of a well-characterized reference material, you can ascertain quite quickly if there's any changes in the growth structure. And this is something that's particularly useful in comparability programs. As with their protein big brothers, aggregation, or the prevention of aggregation, presents one of the major challenges for peptide products. So peptide aggregation is often really very complex and non-specific. However, aggregate formation in itself is really diverse, and we're producing aggregates that vary in size from a dimer to something of 1,000 kilodaltons plus. Unlike the techniques that we've just discussed in kind of sequencing and higher order analysis, aggregation analysis is certainly something that you're going to need to use and apply throughout the manufacturing process. So this is from early development right through to kind of commercialization, and it's perhaps one of the most critical, if not the critical, quality control attribute to actually look at because of the consequence of aggregates and the tendency for them to occur. So aggregates and peptides can be formed either during manufacture or on storage and caused by a number of external factors such as pH, temperature, and even interaction with the container or the stopper surfaces used in the final vessel. In terms of analytical methods to support assessment of aggregation, there are multiple approaches that can be used, including size exclusion chromatography, analytical ultracentrifugation, dynamic light scattering, and NMR. Similar to the secondary order structural programs that you put together, each of these have their own strengths and weaknesses. For example, for methods for longevity and establishing something which can be readily established in most quality control facilities, size exclusion chromatography presents the optimum solution. And with multiple columns used, it can also be applied across a wide mass range, which can be very appropriate for peptides. One potential downside, however, of the SEC approach is that the process of separation can in itself induce aggregation, giving almost false positives, and therefore when you're putting this kind of SEC methods together, careful evaluation has to be made, and a secondary confirmatory tech introduced such as analytical ultracentrifugation, which provides data in situ. Other potential issues to consider is the ability of the method to distinguish between small aggregates that have a really kind of close mass range. For example, unlike proteins, dynamic light scattering may not be the best approach to observe the formation of small aggregates such as dimers or trimers, as you may not get the effective resolution from the monomer. Employing orthogonal analysis is therefore almost a must from a scientific perspective to be confident in the result. But this is also very much a regulatory expectation, with the expectation typically being at least two different mechanism methods be applied. More so, the earlier these methods can be put in place in the peptide program, the better. And when establishing aggregation methods, one recommendation I can really make is that wherever the confirmation of resolution between aggregated forms and the monomer, wherever possible, be proven by using actual aggregates formed through forced degradation as opposed to relying on spike samples of peptides. Perhaps one of the biggest challenges with peptides concerns gaining understanding of their impurity profile. The source of the potential for impurities in the peptides is extensive and encompasses considerable complexity, and important to understand is impurities can have unfavorable safety profiles. Impurities come in one of two categories, product and process related. For process related, identification of potential impurities such as residual solvents, counterions can be made from review of the process itself. And then, if necessary, individual methods applied to measure the residual amounts, with limits for common process impurities such as solvents being really well established. The complexity comes when considering product-related impurities. And in these sort of programs, the first task is to establish suitable methods for monitoring, is to understand the potential routes of degradation. This information is typically derived from false degradation studies, which use LCMS and MES to identify impurities in samples formed after the peptide has been exposed to multiple stress conditions, such as extremes of pH, temperature, oxidative conditions, agitation, light. Once impurities have been identified, the next issue becomes establishing specifications for acceptable limits in product. Here, for peptides greater than 10 amino acids in length, recommendations differ significantly from that associated with small molecules. For those less than 10 amino acids, however, ICH-based regulations can be applied directly. When setting specifications for the larger peptides, the limits need to be considered on a case-by-case basis, based on an understanding of the immunogenicity potential of actually the impurity and the overall clinical indication of the therapeutic itself. Understanding the immunogenicity potential of a peptide is an area that's grown in importance. It is possible to estimate the potential through a combination of in silico and in vitro approaches. which essentially rely on monitoring the interaction of a peptide to the major histocompatibility complexes, or MHCs, the results of which can be directly aligned to the immunogenicity potential. And this is something that's been particularly relevant in the world of peptide generics, where the presence of impurities not observed in the reference list as drugs, but in the generic, have to be assessed via this approach. And there's new guidances that have come out specific to that. The final factor that needs to be considered when putting together a quality control strategy is the effect of the excipients or container system has on the peptide itself. So the objective of formulation of peptide therapeutic is to preserve a stable equilibrium. Peptides, however, can interact unfavorably with some of the preservatives, such as benzyl alcohol, which cause conformational changes to the peptide, or with surfactants such as triene, which lead to the induction of unwanted antibodies. So in choosing a formulation, it's really preferable to avoid using these components. However, if they do need to be included to gain stability or shelf life, additional control analytics may need to be put in place specifically to monitor the effect. In addition, interaction with surfaces or the container or stopper can cause an increased issue such as aggregation. And these need to be considered when choosing the container closure system, but also monitored during stability studies. And this is typically why we perform stability in two different orientations, thereby changing the surface to which the product is exposed. So if we draw those learnings together, the information on this slide identifies the main analytics outside general compendial methods such as pH and appearance that were typically considered in a peptide characterization and batch release stability program. One thing I've not been able to discuss is the need for chiral and optical analysis, but this can be done by kind of standard GCMS or LC approaches. When considering release testing, as kind of mentioned earlier during the presentation, after establishing consistency for certain attributes, the amount of testing can often be reduced, but only after certainty in the reproducibility of the manual project has been established in a really kind of statistically relevant number. The requirement to include a bioassay to monitor peptide activity or potency is perhaps one of the biggest questions that we get asked when developing these solutions. And unfortunately, there's not really a simple answer to this question. What I can say is kind of if a peptide is complex, as is generally in the case of cellular-derived peptides, then the answer is definitely yes. A bioassay is definitely needed as part of your program. If the peptide is simple and synthetic, the bioassay is maybe not so important or required. So thank you for listening. That kind of comes to the end of what I wanted to talk about today. I appreciate it was an incredibly quick overview of the strategy. But for a more deeper explanation, please download our white paper. The information was shown on this slide. And I now can hand over for some questions, I think.
Okay, great. Thanks, Ashleigh. So the first question is, at what stage in product development is testing required?
Really, for some of those key attributes that we've been talking about, it's as early as possible. I think you need to establish really quickly on that your peptide's not heavily prone to aggregation. You need to quickly understand the impurity profile, because if those impurities that might be performed are potentially immunogenic, something's got to be done to control that impurity formation, or perhaps the molecule itself is not suitable because there's going to be so much of that. So really, I think for those key quality attributes, as soon as possible, as soon as they kind of come into the development stream.
Okay. And what is your experience with assessing peptide stability, and when should we think about this?
Again, peptide stability is something that's obviously a formal requirement, so it would have to be done as you kind of progress through the kind of new drug registration process. However, you probably need to get an early on indication of your peptide stability. Inherently, kind of, again, I've said the word aggregation a lot of fields during the presentations, but aggregation is perhaps one of the biggest things that we're looking for in that, to see that the molecule doesn't aggregate. But also we want to check that none of those kind of potential immunogenic impurities are formed. So I think, again, very early on, perhaps as you're going into phase 1, as you would need to for an IND, that's the kind of time that you need to be looking at, and certainly doing kind of mini stability runs as you kind of do your formulation development. The big thing to do as early as possible in relation to stability is to understand that degradation route. So it's not something you can just kind of guess at, and I kind of personally, I suppose, dislike the approach of just throwing every method at it possible. I think what you really need to do is understand it, to do your kind of forced degradation studies, to really push your molecule to extreme conditions, and then use that information and kind of the advanced instrumentation like LC-MS, and even NMR if you need to go to it, to really make sure that the methods that you're putting in place are truly stability indicating and cover kind of all potential routes that your molecule might go down.
Okay. Well, it looks like we are running out of time. We have time for just one more question. If we didn't get to your question, any question that's entered in the question and answer box will be passed on to Ashleigh, and she can follow up with you directly. You can go ahead and continue to type questions in there, but we just have time for this one more during the webcast. So the last question is, sorry, what are the most common questions from regulators about peptides?
Yeah, again, it comes a lot about the things that we're saying. I would guess the most questions that have come back recently are on impurities and making sure that you've actually really well characterized your impurities, and that you've kind of used that characterization knowledge along with kind of modeling of safety potential to actually put limits on it. A lot of what we've seen is the regulators are pretty insistent that you go to the nth+1 or even 10 degree to make sure that you're actually doing that identification as deeply as you can. So this is even in the kind of generics world, when you're kind of looking at marketed products like glucagon and liraglutide, where we're looking at kind of biosimilar type or generic type approaches, whichever kind of application you're doing, we're really having to probably dig deeper into that impurity profile than when the products were registered, so that we really, really understand what's in there.
Okay. Well, thank you very much, Ashleigh.
Thank you, and thank you everybody for listening.
The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Bye.